Well, thank you for the introduction. Again, my name is Pratik Chattopadhyay, and I lead a high-parameter immune monitoring CRO called Talon Biomarkers, and we also help companies develop technologies, including flow cytometry panels. So today, I'm going to talk about panel portability and how we can start to think about translating panels across different platforms, across different flow cytometer types. So what is panel portability? Well, the idea behind panel portability is to adapt panels for use on a new instrument. And we can, so for example, there are many OMIPs that allow us to replicate the work that other people have done. The antibodies will be listed in the panel in the paper for these OMIPs, and we can try to translate these off the instruments that the OMIPs were developed for onto new instruments. And we have some examples here at Talon where we have taken existing OMIPs that were developed on, for example, an Aurora, and we've translated them to our Thermo Fisher Bigfoot, or translated them onto our Sony instrument. And so the idea here is that we want to have mechanisms where we can take existing panels from the literature and then easily adapt them for use on our own instrument. That's the first principle behind panel portability. The second is that when we design panels de novo from scratch, we want to think about making it so that these panels can work on multiple instruments. That's sort of a bigger responsibility that we have as cytometrists. We don't want our work to just be relevant if we're only using a certain type of instrument, because people need to replicate the panels, need to replicate the data. So why does the idea of panel portability matter? Well, instrument differences can really complicate multi-center and team science studies, and increasingly we're doing work in multi-center environments. There may be multiple trial sites where a vaccine trial or a cancer trial is happening, and we need to make sure that we're getting the same results at each site. And increasingly, research is a team science effort where we work with lots of different individuals from lots of different institutions. And harmonizing instruments across those different environments is not trivial at all. In fact, it's quite a challenge. So how have people approached instrument harmonization? Well, one idea that's been implemented in places is to purchase multiple instruments from the same vendor with essentially identical specs. But that's often unrealistic because we're going to do team science with existing resources that are at each of these different facilities, and the capital expense of buying a new instrument is quite dramatic, and so it's difficult to really make it so you plan a study out with matching instruments across sites. Another thing we can do is that we can match MFI signals using fluorescent beads. So we can match the fluorescence that we're seeing on each of the cytometers using, say, rainbow beads or something like that. Many instruments now have an automated routine so that you get consistent signals over time across that particular instrument, and that's very helpful in this regard. And if we line up the different systems across these automated systems and we match the signals, perhaps we can get better at harmonizing. But this doesn't really account for differences in background and noise. And as you'll see in a minute, the differences in background and noise are really what drive whether we can reliably gate on populations and enumerate them. And ultimately, that's the goal. When we're harmonizing panels, we're trying to reliably enumerate different populations and get the gating to match. We can also match the separation of bead peaks. This would capture differences on the low end in noise and sensitivity, but different instruments might just have different background because they have different sensitivities, because the lasers are different, or because perhaps more likely or more important, the detectors themselves are of different and varying qualities, even across the same instrument. We can also measure and match background signals and sensitivity of detectors. This is the idea of using things like an LED pulsar to calculate Q and B values, and then correcting or moving different detectors so that you're trying to match across different instruments. That's often a lot of work and not terribly feasible, but this is probably one of the most reliable ways to go about this, and one of the most comprehensive ways of going about instrument harmonization. Another approach is that we can just forget about harmonizing the instruments and normalize the resulting data. And so that's also an approach that we can try to use. We can try to normalize background levels and normalize signals using bioinformatics, but that often requires bioinformatic expertise, and the algorithms that can be used, differ. You have to make sure you're evaluating them, and it ultimately may not solve dramatic differences, and it seems really risky to do that after the fact and not really set up your instruments carefully. These approaches, so all these approaches that are commonly used, that none of them are really wrong, but they suffer from limitations. They're variously unrealistic, they're labor intensive, or they're not cure-alls. And I don't think there's really a cure-all to doing this, but if we start to understand the components of the cytometry, of the signals that we get in the cytometry experiment, we can start to think about whether we can make this a little bit simpler so it's easier to achieve. So the first component of a cytometry signal is the electronic noise, and this is the noise that the instrument just inherently has when it's running. This varies from detector to detector, especially in conventional cytometry. In spectral cytometry, oftentimes detectors are produced as arrays, and so these arrays of detectors may actually share noise characteristics, and in that case the electronic noise will be similar across many of your detectors. Today's instruments, in my experience, have had consistent and good results for electronic noise. There's very little electronic noise, and it's pretty much even across the detectors, which is really a change from when we were first starting to do this work. Now, we can think about electronic noise, but honestly, the autofluorescence that we get from cells usually exceeds this noise. So electronic noise just becomes a component of the measurement that's swamped out often by autofluorescence. So what is this second component of a cytometry signal? It's autofluorescence. That's just the natural fluorescence of cells caused by different molecules that they have inside them, and it varies by wavelength and by channel. It's usually the worst off the UV detectors and in the lower end of the spectrum. It's typically a minor issue for lymphocytes and PBMCs, but it can really be problematic when you use highly autofluorescent cells that are cell lines or tissue samples or certain other types of white blood cells that can really have a lot of autofluorescence. With conventional flow cytometry, there's not much you can do about this, but with spectral flow cytometry, you can actually get a signal for the autofluorescence that can be unmixed almost as a fluorochrome, and so you can reduce the impact of autofluorescence by unmixing for it, and that's a feature that's available across many cytometry platforms. The biggest component in background, and perhaps the most important part of our cytometry signals, is spreading error. So what is spreading error? Well, spreading error is revealed. It's this broadening of the distribution of the negatives, and that's essentially because of the noise associated with making measurements of low photons and low photon counts. And what happens is that you get this spreading of the distribution that's revealed upon compensation or unmixing. Compensation in the conventional flow cytometry and unmixing with spectral cytometry. You can calculate and you can predict spreading error, but the formula for doing that is non-trivial. I can point you, if you reach out to me, I can point you to the paper where Mario's group, and I was one of the co-authors in it, reveals that formula and talks about that formula. It's not easy to operationalize. Flojo software has actually operationalized it, and some of the other softwares have an ability to calculate spreading error. And so you can try to use that, and we often did use that in conventional cytometry to predict whether we could resolve a marker or not. And that's still a very valuable way of approaching this. Why does spreading error matter? Well, as you can see here, we've stained with CD4 versus CD57 on two colors where there's a lot of compensation, and so a lot of spreading error is revealed. And they're, to make it matters worse, they're far red fluorochromes, and so the photon counting error is typically high. And then to make matters even worse, the signal for the CD4 Cy7PE is really, really bright here. And so you can see that the CD57 positive cells and the CD4 negative population can be resolved relatively clearly and easily. But if you try to examine CD57, what you think are CD57 positive cells in the CD4 population, you see that they're swamped out by this spreading error, by the background. So you can't tell which cells are really CD57 positive versus which cells are just caused by spreading error of the CD4 signal. The population is lost in the spread. So what causes spreading error? You can see how important it is. What is it that causes this, and how can we try to understand that, incorporate that into the idea of panel portability? Well, what causes spreading error, again, is the counting errors that are associated with low numbers of photons that are emitted by some dyes, including those that are in the far red. And another factor can be the poor sensitivity of detectors, but typically your instruments are pretty good nowadays, and so it's less likely that that's the real reason that you have spreading error. What drives spreading error? Well, there are two things really. The level of spectral overlap drives spreading error, because the more spectral overlap you have, the more unmixing you have, and that increases the sort of revelation of this natural spreading error if it exists. Also, the brightness of the overlapping signal is really important. So for example, if we had actually titrated down the CD4, we would see that there might be less, there is less spreading error of that marker then, and perhaps we'd be able to resolve the CD57. In this case, it's likely too dim to resolve, but that's an important way that we deal with spreading error. So the brightness of the overlapping signal matters. It's solved by titrating antibodies and selecting concentrations that aren't unnecessarily bright. So let's review the considerations then for trying to harmonize panels and having them be portable across different settings. The first is that we can harmonize our hardware, right? We talked about that, buying the same instruments, checking the electronic noise. But the issue is that we can't really address this easily. We have to do a lot of lab work to be able to do this, and even hardware work where we're altering the instrument, and it's not really accessible or standardized. And electronic noise probably doesn't matter all that much for panel portability, because the other issues like autofluorescence and particularly spreading error are more important. Well, let's talk about autofluorescence then. Does it matter for panel portability? Well, oftentimes we'll see that our panels have more spreading error than the autofluorescent signal, and so it's less of a problem. Also, you can unmix the autofluorescent signal, and if you're working with PBMCs like we primarily do here at Talon, it's just less of an issue. You don't have to deal with it so much. But when we talk about spreading error, that is something that's an issue and that can be dealt with easily with minimal bench work and in an accessible, standardized way. There are two ways that we can deal with spreading error. We can lower the brightness of signals by titrating down our antibodies, and that's relatively easy and is trivial to do, and is probably within the best practices of how we should be doing high parameter flow. And then another way to look at it is thinking about the spectral overlap, because if we can minimize the spectral overlap that we have, we can minimize the revelation of this spreading error. So how do we quantify spectral overlap? Well, in conventional cytometry, we can actually do this experimentally, right? We can stain compensation tubes, we can build the compensation matrix, and then we can look at the intersection of each of these dyes to get a relative reading of how much they overlap, right? If the compensation value in the matrix for FTSE versus PE is very high, then that has a lot of, if that value is very high, then there's a lot of overlap between those dyes. In spectral cytometry, we don't really need to perform an experiment like that, but the math that's involved in calculating the complexity and the spectral overlap is different, and it's more complex. So if we're going to do panel building, It typically depends on the platform that you're using. So Aurora has a system, the Cytek Aurora has a system where you calculate similarity of dyes, and there's certain similarity thresholds that that they think will work versus what doesn't seem to work. On the Thermo Fisher Bigfoot, they calculate complexity on board on the instrument, and you can use that to plan out your panel. Again, though, the underlying math is complex, and one of the things that I want to communicate in this talk is to make all of this simpler and easier to do. And so how is it calculated on the Bigfoot? Well, each dye's spectrum is essentially represented as a vector, and the dyes that are very different essentially have a wider angle in space when they're arrayed out in space. And dyes that are similar have a narrower angle, a more narrow angle in space. And so the complexity of the panel is the sum of these different angles, 1 over the cosine of all these angles. When I see something like a cosine or a sine, I lose my mind. So I started to think about how to do this in a hopefully more simple way. And what the challenge is right now is that we have to go to the instrument in our workflow and build our panels out, and then use that proprietary software, look at the complexity or the similarity scores, and then go back and say, okay, well, what if I replace this reagent with this reagent? Does that lower the complexity? And I have to keep doing that iteratively, which is kind of a pain to do. And so today I'm going to show you how we may have developed a better method, and this is what we are currently fine-tuning and working towards publication with. The method that we've developed is much easier. It doesn't require any bench work. The math is simple and accessible, and the workflow doesn't require iterative trial and error on proprietary software. So the underlying principle here is that we want to design a panel that will work across multiple instrument platforms so that we can reduce spreading errors by choosing dyes that don't overlap as much. In recent years, we've really had an explosion of new dyes, and so there's an enormous number of choices in terms of the dyes that we could be using. So how do we smartly choose those dyes, and how do we incorporate that into the idea of panel portability? That's the remainder of the talk. So when we're choosing dyes, we can almost think of our panel building process in 2 different ways. In some cases, we're trying to pack panels where we're maximizing the number of markers. And when we're trying to do that, we're looking for dyes that are similar but slightly different in spectrum, and different enough that they can be unmixed. And we choose markers to investigate on those that are maybe exclusively expressed by different cell types, and so on. And that's really when we are trying to put the pedal to the metal and do the coolest, biggest panels, right? That's what we're focused on. Spectra that are just ever so slightly different in order to pack in as many of those as possible. If we're thinking about panel portability, though, and we're not trying to do the coolest experiment, and instead we want to make sure we have the most robust experiment that can easily work across different instruments, then what we want to do is do something a little bit different. We want to choose the least spectrally complex dyes so that overlap is less likely, and therefore spreading error is lower. And I think that is the recipe for success here. So our method basically starts with downloading the reference spectra. So we go to sites like FluoroFinder, like BD Spectral Viewer, like Thermo Fisher's Spectral Viewer. In this case, I use Thermo Fisher's one because I could download the spectra and just plot them on my computer. And what I do is I download these spectra, and then I try to start to fill the panel with the least complex dyes. So how do we identify the least complex dyes from the downloadable spectra? Well, here we've got 2 examples, NovaFluor Yellow 810 and PE size 7. And this is just the emission that comes off of these reference spectra. And when we plot the raw spectra for the dyes, it's kind of possible to see which dye has a broader spectrum and thus has the potential to overlap with more dyes, but it's not as easy as it could be. So what do? We transform the spectra to make the differences more dramatic. And then you can see here on the right side that the transformed spectra really reveals these differences. So essentially the way we do this transformation is we replace any non-background emission signal with a value of 1, and then everything that's background is 0. So we just, amplify the differences basically. And so you see this little shoulder here gets amplified, and the difference between that little shoulder doesn't really matter to us. What we're looking for is essentially the breadth of this peak here. And so you can see that Nova-4 Yellow 810 has a less wide peak than PE Psi 7 for its major peak. And even for the minor peak, it's smaller than PE Psi 7. And because of that, it's likely to have less overlap with other dyes. So we would choose to use that dye in our panel. And we can actually sum these zeros and ones across the entire spectrum to give a dye simplicity score for each of these dyes. In the case of Nova-4 Yellow 810, that score is 188. In the case of PE Psi 7, the score is 252. And when we do this we did this for 101 dyes that I could think of off the top of my head. And I did this for all the lasers except the UV laser, because a lot of those dyes, there are a lot of choices for off the UV. They're mostly brilliant UV dyes, and many of them are going to be less complex just because there aren't that many places for them to spread into. But we can repeat this, and we will repeat this with the UV dyes. So what we've got here is the emission wavelength of a particular dye. The letter indicates what laser it's excited by, and then the dye simplicity score is plotted on the x-axis. And this is a nice reference that you can start to use to build panels. This reference is instrument agnostic, and it's based completely off the dye spectra that we get, that are reference spectra that come from the people who make these dyes. And when we start to look at these dye simplicity scores, you can see the big differences, right? Here's Q.605, and here's our transformed graphic for the spectra, and you see how narrow it is compared to this dye, Nova-4 Blue 690, which is very, very broad in its emission. And so we can start to see some interesting insights here. The first is that this set of these that have the smallest dye simplicity scores are actually quantum dots. And we know that quantum dots have pretty narrow emission spectra, but it's remarkable how much more narrow they are as a family compared to other dyes. And I realize that quantum dots have kind of fallen out of favor in panel building, but perhaps we should think about them in settings where we want to make sure the panel can be used in different across different instruments. The other thing to note here is that there are some gaps or some barren areas in this plot. From 450 to 500 nanometers and from 700 to 800 nanometers, there are very few low complexity dyes. And vendors and chemists who can come up with lower complexity dyes in these regions, these dyes would have a lot of value to us. And so it gives dye developers some ideas of what's worth developing that would be truly novel. So when we want to use this graphic to build simpler, less complex panels, what we want to do is split this graphic into thirds. And so we can build a minimally complex 5-laser 17-color panel by cutting the dye simplicity scores into thirds. And then what we can do is we can basically start filling the panel with the simplest dyes in the first sector. We can select dyes that are about 25 nanometers apart. And so we can say, okay, I'm going to first think about 400 to 425. This dye that I'm highlighting, hopefully you can see my arrow here, 400 to 425 that has a dye simplicity score around 125 is a great choice there. And then we can continue to build up, look in the slice between 425 and 450 and go through that. And then we come up with a list of 17 dyes that are the least complex and that would likely work together quite easily and well in a panel. And this is regardless of instrument type. Again, I can't stress that enough, because this is just from the dye spectrum. So you can just use this list and start to build 17 color panels that way. the sum of the dye simplicity score for each of the dyes. So if you sum these scores for each of the dyes, you get a number, and that can actually tell you the measure of panel port. That can give you a measure of panel portability. So if we choose dyes that are only in this, or mostly in this first sector, where the dye simplicity scores are the lowest, the sum is 2,111. If we choose the most complex ones, if we choose the ones that are in this third sector of the dye simplicity score graphic, then we get a much higher value, which indicates that the panel might be less portable. So what do we need to do now? We need to go back and actually build out these and show where the differences are and why this matters. We're working on that. I'll talk a little bit more towards the end of the talk. We can also, once we've done this, we can actually organize the information for our particular instrument. So what I've done here is I've put on the x-axis all of the detectors on our Thermo Fisher Bigfoot, and you can see B1 through B7. Our blue laser detectors, R1 through R5, are the red laser detectors, and so on. And I can organize the dyes by the detectors where the max emission basically falls within the filter set for that dye. And you can see now that I can start to compare these different dyes, and I can ultimately choose the ones that have the lowest dye simplicity scores to build panels to maximize out across our single instrument. And again, this is with downloadable spectra. There's no investment of reagents that's required for this. And so using this method, I can actually name out what the best dyes are on the Bigfoot, on the Thermo Fisher Bigfoot, and there's a list of them here. Now, does that mean that we can't use the dyes that have higher complexity scores, or that we, or that the other dyes are less valuable in any way? No, of course not, right? Because this is just a simple panel that, we can port across different big feet, I guess. But there's no reason that other panels that substitute out some of these dyes, especially ones that are close in the simplicity score, wouldn't also work. The other thing that we're not considering here is what markers are available on these dyes, and we're not considering some of the characteristics of these dyes, which could be important as well. Like some of them play well with intracellular staining while others don't, and that's not folded into this. And so there's certainly more consideration that you have to do. We have another instrument here that's a conventional 12-color flow cytometer, and I just wanted to highlight that we can pick out the best dyes for that instrument as well, and that they might differ from the ones for the Bigfoot, of course, because the filter sets are different. And so when we think about a real-world scenario, as I mentioned at the beginning of the talk, we did this with an OMIP that was built on an Aurora and needed to be translated to our Bigfoot. And this is an example. Actually, I'm not giving an example of what we've done. This is a possible real-world scenario. So this is where OMIP 84 that looks at gamma delta T cells built out this panel using Cytek Spectral Viewer. They recorded similarity, and then they had to iteratively choose dyes until they worked. Well, we wouldn't have to choose that, because what we could do is just pick out the dyes that are likely to work from the start based on their dye spectrum. And that's like a first baseline pass. So it might make panel development much easier without multiple interrogations. And so if we look at the dyes that are used in that OMIP 84, and then we compare to what simpler dyes would be, would essentially fluoresce in the same or neighboring region and could be used in the panel, we actually see that there are a lot of other choices that could be used. And so if we had a project where we as a CRO needed to develop a gamma delta T cell panel, excuse me, and we needed to use it across different settings or different places, the first thing that I would do is go to the excellent work of this group, where they've reported out a panel that works, that looks beautiful, that I can look at the graphics for the staining for and understand what I should expect. And then I would try to translate out these different dyes that they use into simpler dyes that would perhaps make it robust and easier to use across different settings. The most likely improvements that we would see would come in the far red channels, where the low photon counts result in higher spreading error. And there are two important caveats here. Our system doesn't yet weight dyes by their brightness, so a simpler dye could still give too dim a signal to be of value. And then the other important caveat is that some of the marker dye combinations may not be available for the things that we are proposing as simpler dyes. For example, dye light 650, you probably have to conjugate a reagent yourself in order to, an antibody to that yourself in order to use it. We might also consider expanding our panel on our Bigfoot by adding markers that are on neighboring channels and with dyes that are more simple, because those dyes with simpler spectra are easier to use when we're trying to pack the panel. And so if we were to go in and want to pack this panel, we would add dyes off, in many cases, NovaFluors, like NovaFluor Violet 745 or a series of NovaFluor Blues, in order to get more out of this panel. So to summarize then, what I've talked about today in terms of the key concepts is this idea of panel portability, transferring panels across instruments. And if you had asked me about this 10 years ago, 15 years ago, I'd like, that's a lot of work. And I'd say that the first thing you have to do is harmonize your instruments. But a case could be made that if you're transferring panels across facilities that have relatively modern instruments, you may not have to do the difficult process of instrument harmonization, which might not be feasible or can be challenging and imperfect. Or maybe you do a little bit of instrument harmonization and then you make sure everything works by choosing better dyes. And with the availability of many new dyes, we may be able to curate panels to reduce spreading error. Panels that use dyes with narrow spectra that consist of a single peak may be very easy to transfer, more robust for long-term studies, and really importantly, easier to gate. Current panel building tools require us to build a theoretical panel, assess its complexity, then compare that panel to iteratively rebuilt panels that substitute different dyes. And we have staff who go to the Bigfoot and basically sit there for an hour and fidget with different combinations of different dyes and look to record everything and look to reduce the complexity. That can be cumbersome. And so we would propose that comparing the spectra of dyes is a better way to do it. The problem with comparing raw spectra, though, is that each of the spectra are unique. They're continuous. They have shoulders all over them. And the math involving comparing spectra and panels is cumbersome. So we propose, as an innovation here, to do a simple transformation of the publicly available spectral data to allow easy comparison of the dyes alongside simple metrics that could be applicable for dye and panel comparison that don't require too much thought and certainly don't require any cosines or tangents to work. The limitations are, of course, that we don't account for dye brightness, and this can vary by instrument. So our approach is a little too simplistic, and we need to have some shades of development there. We're developing a simple approach to assess differences between dye brightness and incorporate these into the algorithm for panel design that we use. We can't predict antibody clone performance. Really, nobody can. You may have different clones of antibody that look better or worse depending on the, even though they're on the same color. We haven't, but can pretty easily match markers to dyes suggested by our approach. So based on whether a marker is exclusively expressed, whether it's on-off expression or dim or continuous expression, we can start to factor that in as well and start to match up markers and dyes. It's not terribly difficult to do. And we don't really have data yet from panels that are built with and without our system. We're in the process of doing that. I would make the argument that there are so many factors that go into a panel working or not working. Perhaps showing you that it works for one particular panel isn't critical. I think what's more important is that we stain out CD4 on cells or on dyes using the same different or on beads using different the same clone on different dyes, and that we build out different panels and look at their spreading error. And that will really tell us a lot about whether this system is working or not. One of the things that we have done is compared it to the complexity that is calculated by, for example, the Bigfoot. And we're finding that our system with simpler panels correlates very well with complexity that Thermo Fisher system calculates, but with larger panels that we think are really complex, the Bigfoot sort of sees them as less complex. So there's a disparity there that we can start to get at with some experiments. But overall, a major advantage of this approach is that we can build it into our workflow without actually doing any bench work. We can plan out panels in dry space. There's no hardware assessment or capital reagent investment required. We reveal which dyes amongst the synonymous options, the ones that we see using the same detector, the same filter set. We can pick out which ones are best on our instruments, again, without doing an experiment. We reveal where dye developers have opportunities to introduce improvements or new dyes, and this is an easy way to translate OMIPs to give better resolution that's customized for your particular instrument. And I just want to highlight that this is the type of work that we're doing at Talon. We set a very high standard for ourselves in terms of innovation. We are developing automated, evolving, simple panel design approaches so that we can build panels faster and easier and really lead the industry in that. We have a broader suite of automated QC tools that I'll talk about sometime that allow us to do the quality control that I used to do manually, to do that in an automated, fast fashion. So you're not waiting for that data, and you can catch errors when they're still correctable. We rapidly translate panels across instruments, and that also improve panels to address the client's particular focus. And then we have a number of new initiatives that I'm excited for you to reach out to me to talk about. These include efforts in leukemia and lymphoma. They include the idea of being a one-stop shop, which we now have all the equipment and experience for, to do spatial work, to do flow cytometry, and to do plasma proteomics as well. And then we're developing data analysis approaches also that help to maximize the information that we get from the data. And so you can reach out to me, Prateep at Talon Biomarkers, if you want to talk more about panel development, anything in cytometry, or to learn more about the services that we offer. You can also get on the waiting list for our new tools. And our goal really here is to advance the science. I want to thank Alex, who's our operations manager and senior scientist in the lab, Mitra, who is an intern who is developing some of the interfaces that we can use to do this easily. And of course, whenever we're doing this work on patient samples, we're very respectful of and thoughtful about appreciating the dedication of people donating samples. And so at Talon, we assay every sample type from a study, combining data from each modality to improve discovery and biomarker specificity and sensitivity. And we do that alongside technical development and data analysis as well. So I will leave it at that. And we definitely have some time for questions. Thank you all for paying attention and your interest.